Corynebacterium diphtheriae. Now, Corynebacterium diphtheriae is actually a gram-positive rod which is V and L shaped. Now, the particular feature about this rod is that it has no motile ability, meaning that it is non-motile organism. Another beautiful thing about that is that you can see specific red granules. Now, these red granules can only be seen by a stain known as methylene blue. Now, why is this visualized? Because this is actually the granules that is composed upon polymerized phosphate bonds. These polymerized phosphate bonds, when exposed to methylene blue, turns red, while the rest of the organism stays blue. So this is one of the differentiating features in this organism in order to differentiate between the other similar organism which is V and L shape, which is known as Listeria monocytogenes. Now, how it is this organism caused? Now, this organism results in a condition known as diphtheria, hence the name Corynebacterium diphtheriae. Now, what happens is that this organism enters by air droplets. When it enters by air droplets, it is attached to the oropharynx, especially to the tonsillar area. Now, upon attaching to that epithelium, what happens is that we have actually two types of this organism. The Corynebacterium can be virulent and can be aviral, meaning that some strains can cause disease and some strains don't cause disease. The strains that do cause disease are the virulent ones. How are they virulent? Because before getting into the body, they are lysogenized by bacteriophage, meaning that they are visited by a specific virus which consists upon a specific DNA which is encoded into the bacterial DNA. Now, this virus is known as a beta phage. Upon encoding its DNA, it makes the DNA produce a certain toxin which is known as diphtheroid toxin, which actually is the main thing that causes the disease. So, when this organism secretes a toxin known as the diphtheria toxin, this diphtheria toxin, as you can see, consists upon two parts. A part is the attaching subunit. Now, this secreting part, if it is being attached to this part, A part will attach to the binding subunit, meaning that it must have a specific type of receptors with which this toxin actually binds. And then it is a B part. The B part is actually responsible for enzyme induction. Now, it results in a specific enzymatic reaction. Now, the specific enzymatic reaction which takes place because of this AB subunit is that it converts nicotine amide into adenine dinucleotide. Now, it is actually affecting some enzymes which are present in the nucleus. Now, affecting the nuclear DNA of that epithelium. Now, by making NAD, what happens is that this NAD actually inhibits the DNA by forming a messenger RNA that can be readily to be formed certain type of protein. So if by inactivating it, it is inactivating actually the elongation factor 2, which is the thing that is supposed to which is supposed to read this DNA in order to form the messenger RNA. Similarly, the cell protein would not be formed and the cell would similarly die. So in the next stage, what happens is after this elongation factor has been inhibited, what happens is inflammation occurs, that our neutrophils, our leukocytes, all of the WBC come to this respective epithelium, and it causes fibrinoid type of degeneration, which give rise to a gray colored pseudomembrane that is actually present in the oropharynx or over the tonsils, which upon removal can result in a little bit of a bleeding. So we should not obviously remove it. Now, this is a gray colored pseudomembrane which is formed in the oropharynx, which can be formed over the tonsil and even in the throat. So what are the symptoms? If it actually covers the throat, it can actually result in serious complications such as airway obstruction. It can even go to the heart and cause myocarditis. In the CNS, what it causes all cranial nerves which are actually present paralysis. And it can go, even if this is closed, obviously if the larynx is being affected and there is laryngeal pseudomembrane form, all of the food that is actually going from the oropharynx would come out through the nose. Now the specific thing is after a few days to weeks, antibodies are formed against this organism which is present over here. Now the antibodies can be assessed by, or the toxin can be assessed by a test known as Shahik test. Now Shahik test is what happens is that we give 0.5 ml of standardized toxin that is given intradermally under your dermis, and if the antibodies are formed, if the antibodies are positive, there is no inflammation, and if the antibodies are negative, there will be inflammation because antibodies would be formed respected to this toxin, but if it is already present, it would not result in any sort of inflammation. This inflammation normally occurs up to 4 to 7 days. Now, the laboratory diagnosis of this organism is that we use a gram stain, and our gram stain will tell us that there is a V and L-shaped rods, and by using methylene blue, we can differentiate it from other bacteria which are also V and L-shaped, such as Listeria monocytogenes, because upon methylene blue, it has reddish colored granules because of the phosphate bonds. Now, this organism especially grows in a specific medium known as Loeffler's medium. And this Loeffler medium is specifically has a tellurite plate. Now the tellurite plate consists of a tellurite sol, which this organism actually reduces. Now if this organism actually reduces, meaning that it is using it, so the organism gets tellurite on their colonies and they become blackish. Now, another test for quick diagnosis of this organism, because diphtheria is very fatal disease, what we can do is we can do PCR, which checks for DNA, and we can do a precipitin test for checking of the antibodies. Now, the treatment for this is, the organism itself is not dangerous, but we are we are actually depressed about the toxin that is produced. So what we give the patient is we can give the patient antitoxin first, so in order to avoid serious complications, which is diphtheria antitoxin, and for the organism we can give the patient penicillin G and erythromycin. Prevention is that we give the newborn babies TTAP vaccine across 2 months, and then after at 11 to 12 months. So in order to get sufficient amount of antibodies within the organism to prevent such a serious complication, this vaccine is combined with tetanus and pertussis vaccine.